Good evening, everyone. Welcome to another session of Botany Insider's free session. Right, we'll be learning about new concepts. We'll be revising new concepts. One of them is your nucleosome concept, and the other one is your transposons. Right, so we'll be learning about both of these, revising about both of these concepts. Right, let's begin with the class now. If you haven't yet downloaded our application, so the link for the same is in the description box. You can click on the link and get yourself registered on our application, and you can access a number of courses that are running. If any of you is targeting the June 2024 examination, so we have a full course running for the same. We started this particular course on 17th of November. So any one of you who wish to enroll in this particular course, you can download the application and get yourself enrolled in the same. The pricing for the same is 4949 only, right? Let's begin. In addition to that, we also have a mock test series going on for the upcoming examination. You have just 25 days left for the upcoming examination. So, this mock test series will help you out in practicing out the questions with the best of your ability. Right? Let's begin with the class. Good evening, Malik. Right? Let's begin. First, we'll be discussing about the concept of nucleosome. Then we'll try to solve out few questions as well. Then we'll be learning about the second concept, which is your transposons, depending upon the time. Okay, let's begin. First, let's start with what nucleosome is. So, if you look into the previous year questions in 2023, also there was a question asked, and it was based upon the zigzag model, and it was based upon the solenoid model. In addition to that, you also get questions from the organization of the genome, the organization of your nucleosome. So we'll be learning about that as well. Let's begin. So what is a nucleosome all about? Nucleosome basically means it's a repeating subunit of the chromatin that are packed inside the cell nucleus. See over here, this will give you a better picture. So, in the nucleosome, what do we have? The nucleosome is completely composed of your histone proteins, right? In the histone proteins, you have two types of histone protein. One is your core histone, and the second one is your linker histone, right? Core histones are the one in which we have H2A, H2B, you have H3 and H4. All of these are arranged in, or all of these are in two units. So, 2 H2A unit will be there, 2 H2B, 2 H3, and 2 H4 will be there. The linker histone is the H1. The variants of all of these are available in nature. So, remember about this as well. All of these get organized in the format of an octamer, right? Remember, all of these get organized in the format of an octamer. Now, after the octamer is formed, then we see the base pairs. We see number of base pairs getting linked together with the help of this orientation. So let's say this is a thread and this is your octamer, right? So that's why we call this nucleosome as beads on a string structure. These two of the octamers that we have, they get pulled towards each other with the help of a linker histone, which is your H1 histone. So H2A, H2B, H3, and H4, these are organized in a format that they'll be making up a histone octamer, and then the linker will be helping in joining of these octamers together so that the packaging can increase, so that we can see the condensation of the entire chromatin fibers, and finally you can check or you can see the chromosomes in the existence. See over here. So you have initially in the cell, in the early interphase, you have your portion of the interphase chromosome. In this, what is an interphase? We have discussed about this yesterday. What is it all about? So you have G1 phase, S phase, and the G2 phase, right? So these are three parts. These three are the parts of your interphase. So in this particular time, we see the chromosome in the format of a histone octamer, right? So in the format of these chromatin fibers. Now these chromatin fibers first gets converted into 30 nanometers, then we have 10 nanometers, then you have 2 nanometers. So condensation is increasing at every single step. So you have these chromatin fibers this way. If I look into this smaller segment, this is the this is how it looks like, and then you look into it in the DNA format. So the DNA gets condensed in the format of your nucleosome fiber, beads on a string structure. This beads on a string structure then gets converted into the solenoid. Now this solenoid is what? This is the structure. That's why there are two types of model which explain this. One is a solenoid model, other is a zigzag model. What is the baseline difference between the two? We'll just understand. Now this solenoid model, when I look into the most condensed format, so this is the chromatin fiber then gets arranged in a way that you see the chromosome in the nucleus. So you have to remember about each of these pointers. Very important, very scoring, and also logical as well. If you go in a point wise and in a sequential manner, you won't find a difficulty in understanding this, right? See over here, you have the double helix DNA 2 nanometers, then beads on string structure is 11 nanometer, then you have 30 nanometer, which is the solenoid model or the solenoid format. Then we have 300, which is the section of the chromosome in an extended form. Then we have 700 nanometers, that is condensed section of the metaphase chromosome. And finally, 1400 nanometer condensation, where we have the entire metaphase chromosome in the orientation, which we look into our main DNA, right? So DNA is like this. Chromosome is what? It is the condensed format of your DNA. We have various levels of condensation going on, and this condensation is based upon the nucleosome. What is a nucleosome? We have just understood. How? What is it made up of? Made up of linker DNA, linker histones, and your core histone. Core histone consists of four of the types: H2A, H2B, H3, and H4. And then you have the linker one. So linker is based upon, or it is your H1. H1 is the linker. All four are your core histones, right? There are two major models for the same. One model is zigzag model. Other model is your solenoid model. The solenoid model says that you have the chromatin fiber rolled or arranged around a central axis. So you have a central axis, and around this we have our final arrangement of this entire chromatin model. Zigzag model says that there is interaction between the nearby alternate nucleosomes or alternate beads on strings that are getting formed. So the solenoid model is a more accepted model. Zigzag model is less accepted in nature because it is not fulfilling the exact information, or it is not fulfilling the exact scenario which we actually see in nature, right? That's why the solenoid model is much more in existence. What is happening in solenoid model? It says that you have the central axis, and around this central axis we have the chromatin fiber getting rolled around in the format of beads on a string structure. And the zigzag model says that we have our chromatin fiber in the zigzag model with the pairs or with the alternate nucleosomes, histones, segments that we have, they are interacting with each other. So remember about this segment as well. Now see, this was a question that was asked in June 2022, and it is related to your structure of 30 nanometer chromatin fiber itself, right? Let's understand this question. First option is, in the solenoid model, the linker DNA connects the consecutive core histones. Second is saying that in the zigzag model, alternating nucleosome become interacting with each other. Third, in the solenoid model, 12 nucleosomes are arranged or organized on separate stacks, while in 8 nucleosomes per turn makes up a single zigzag helix. Then you have the H1 histone is essentially required for the zigzag but not in the solenoid. Last option is chromatin fibers prepared with your H4 histones that lack their tails could fold into higher order fibers. Now you had to find out the combination of the correct statement. It's a very, very logical question. It's a very easy question if you know the facts behind what is a solenoid model and what is a core and what is a zigzag model, right? Let's try to solve it out. Now, first one is in the solenoid model. What we have just studied in the solenoid model, you have the central axis and then the coiling getting done. Then you have the zigzag model in which you have the consecutive nucleosomes are getting or they are interacting with each other. So, first option, the linker DNA connects the consecutive core particle. A option is correct. I'm sure about A option. Then in the zigzag model, alternating nucleosome become interacting neighbors. This one is also correct. Okay. Then you have the third one that in the solenoid model, 12 nucleosome are organized into two separate stacks. It is saying that in the solenoid, it is arranged in two separate stacks. C is wrong. I'm sure that C is wrong. Then H1 histone is essential only for the zigzag and not for the solenoid. This one is also wrong because it is required in both of them. Because for us to get both of the alternating or both of the core histone octamers linked to each other, we require linker DNA, right? We require linker proteins for the same. Then, so C is wrong, B is wrong. If I keep eliminating these things, C and D being wrong, this is not the correct answer. C being wrong, this is not the correct answer. Now I'm sure about the first and the second option. So either A, B is the correct answer or only A and B is the correct answer. Fourth option is last option is saying that chromatin fibers prepared with the H4 histone that lack their tail could fold into higher order fibrils. H4 histones does not have tail to them. It is the H1 histone which is the linker one which have tail to it. So E option is again wrong. I can eliminate option number B as well. And the combination which is correct is A and B only. So C is the correct answer for this particular question. Only A and B statements are correct. All other statements are wrong. If you have paid attention to what I have told you in this particular slide, you can easily solve out this question. You should know the facts behind what does solenoid and zigzag model is, and you should know how to solve the question with, and this can be done or you can learn this only if you practice out the things properly, right? Easy question, simple question. It was a part C question, that is a 4 marker. Is it clear to all? Yes or no? Just check whether the question is clear to you or not. I've explained you the concept also, and I have given you the entire question detail as well. Yes, just check whether it's clear or not, everyone. If it's clear, please let me know in the chat section. Then we'll proceed forward to the next question and the next concept as well. Yes, done everyone? Clear? Perfect. Let's move forward. Right? Let's do one more question. Okay, see over here. Following statements are made with reference to the structure of the nucleosome. Okay, again, structure of nucleosome is given to us. You need to find out which of the following combination represents all the correct statement. Whenever you read a question, and this is from June 2021, okay, in last 3 years consecutively we have got question from nucleosome concept. Okay, so do not skip this particular question at any do not skip this particular concept at any cost. So first option is a histone tetramer is a core of the nucleosome comprising of two H2A, H2B, H3, and H4. Then the N-terminal tail of the core histone are believed to stabilize the 30 nanometer fiber of the nucleosome DNA by their interactions with adjacent nucleosome. Then post-translational modifications of the N-terminal tail as well as the globular tails of the core histones modulate the transcriptional events. Then, according to the zigzag model of the 30 nanometer fibers, the linker DNA circles around the central axis of the fiber as the DNA moves out from one nucleosome to other. It is talking about in the zigzag model, you are having a central axis. D option is absolutely wrong. So if I'm sure about D option being wrong, I can eliminate third and fourth. If you look into the first option carefully, it is saying histone tetramer. What did we just study? It's a octamer, right? Octamer is 8 units. So H2A, H2B, H3, and H4, all of these arranged in 2 to ratio, right? 2 ratio each. So first option is also wrong. I can eliminate first option as this option as well. So B and C options are correct. So A is the correct answer for this particular question. Again, easy scoring and very, very logical question. If you know about the zigzag model, if you know about the complete nucleosome concept, you can easily get your 4 marks in hand. Is it clear to all? Yes or no? Just check whether it's clear or not. If yes, we'll move forward to the next question. Yes. Go through the question. It's very, very simple, very easy scoring question. Even if whatever I've explained you in this class, even if you keep only this particular point in mind, you won't find a difficulty in solving out the question in the examination. Okay, let's move forward to the next question now. And the next concept now. Okay, now we'll be revising about the second concept of today's class, that is your transposons. Okay, we'll learn about what transposons are. So, what are transposons? Why do we study them? What is the entire concept behind the same? Let's understand about the same. Okay. So, what are transposons all about? We learn about transposon in a way, or we call them the jumping genes, right? What are these? These are the jumping genes, the ones that keep on jumping from one side to the other side in the genome with a specific functionality, obviously, right? So, let's see and let's understand about the same. Yeah. So, what are transposons all about? So, what are transposons? Simply, these are your jumping genes, right? We have various classes to it. We have various other concept to it. I'll give you an entire I'll give you an entire list to the same, one flow chart for the same, and then you won't find any difficulty in solving out any question from this segment specifically, right? Let me write down the entire list for you. So when we talk about transposons, in the transposons you have two major classes. In this you have retrotransposons. You can keep this particular flowchart with yourself, it will help. And on the other side, you have the DNA transposons. Okay. In the retrotransposons, you have two subclasses, LTRs and non-LTRs. In case of LTRs, there are two examples. One is TY, which is found in yeast, and the other one is copia, which is found in your Drosophila, right? Then you have non-LTRs. The non-LTRs have again two of the subclasses. One is your sine other one is line, right? So in the format of your sign, there are three examples. One is your ALU, other one is BLU1, and then you have BLU2. ALU is found in humans, and BLU1 and BLU2, these two are found in mice. Then you have the example of line. So in this, you have L1, you have R2, and you have I. L1 is found in humans, R2 is found in bombox, and then you have I found in Drosophila, right? Then you have DNA transposons. These are your second class, class 2. In this, you have two further subclasses, like two examples. One is the P element, and the other one is ACDS. P element is found in Drosophila, and ACDS is found in maize. All these are important, okay? Keep a note of this entire flowchart, and you will never face a mistake. You will never face a problem in solving out the question for your entire transposon concept, okay? So this is your entire list which you can keep a note of. So transposons, two classes, retrotransposons and your DNA transposons. Retrotransposons, again two classes, LTRs, non-LTR. LTR, two examples, TY and Copia. TY found in yeast and Copia found in the Drosophila. Non-LTRs, sign and line. Sign found in ALU. Like signs example are ALU, BLU 1 and BLU 2. BLU 1 and BLU 2 is mice. Then you have the sign. Example is L1, R2, and I. L1 is found in humans, R2 bombax, and then you have I found in Drosophila. DNA transposons, these are the second class. Examples are uh, P element and ACDS. P element found in Drosophila, ACDS found in maize. Remember about this, very, very important. If you are like completely sure about all of these pointers, then no one can stop your 4 marks or 2 marks, whichever gets asked in the examination. If you look into last 3-year papers, every year we have got a question from transposons, okay? In 23, 22, 21, all the years we have got questions from the same. Let's try to solve out few questions related to the same, everyone. So this is the first question. See over here, you have LTRs, non LTRs, and sign. I have just told you the same thing. What have you learned in sign? Signs example is ALU, BL1, BLU1, and BLU2. So C gets marked with the third one. C getting marked with third. Then you have LTR. So LTR is TY and Copia, Copia element. So non LTRs, LTR is copia element. One gets marked with second, and non-LTR, you can keep a note. Over here you have 7 to 21 base pair sequences. I've just told you the exact same thing. See over here? Just a second. Where is it? Okay, anyway, so you have the answer as option number 1 with the flowchart with which I have just drawn for you, right? Same, we got one more question from the same. Before that, just go through this if it's clear with you all or not. If yes, then I'll proceed further with other topics. Very important, very, very easy, very scoring as well. Just keep a note of that particular flowchart and you won't find any doubt, any problem in solving out such a question. Okay. Done, everyone? Clear? Understood? Yes or no? Just let me know in the chat section. Yes. Is the question clear to everyone? 8 of you are there in the class. Please check whether it's clear or not. See, this was one more another question that was asked. This is a group of transposable element described as retrotransposon encompasses which of the following? First one, P element in the Drosophila. Second is line but not sign in the humans. Third, you have copia element in the Drosophila, sign but not line in humans. Then you have copia element in Drosophila, line as well as sign in the humans. And last, you have the P element in the Drosophila as and line and sign, both of these in the humans. So if you understand the question properly, C will be the correct answer for this particular question. We have just studied copia element is there in Drosophila and line and sign, both of these are there in your humans as well. So C is the correct answer for this particular question, right? So the flow chart which I just gave you will help you out in solving out all the questions that we have done today, right? So I took up very important concept, two very important concepts today, right? Be there in the class every day, every single day till your main examination. We'll be having our classes on YouTube channel, same timing, 8 o'clock every single day, right? This will help you out, and I'm trying my level best to provide you with the best and the most useful content possible for your upcoming examination, right? And if any one of you is preparing for the June 2024, we have started off with our course for the June 2024 as well. If you wish, you can enroll in this. The link for our course, the link for our app downloading, every single thing is there in the description box. Check it out, and if you wish to enroll in that, you can download our application and get yourself enrolled, right? So tomorrow we'll start with the next unit. We'll be again taking up few topics, revising them, solving out questions as well, so it will help you out. Okay, so be there in the class at 8 o'clock and try to solve out questions with us and revise with us in the best manner. Okay, bye everyone, take care. And after, like, those of you who will be watching up the recording, if you feel like that you want help in specific topic or something, you can let us know in the comment sections also. We'll try to make up a class, or we'll try to arrange up a class accordingly. Okay, all the best, bye everyone, take care.